from the chapter on viruses. So people are, well, what do I need to know about viruses? So viruses, as you realize, are incredibly different. There's so many different ones of them. So you really want to make sure you pay attention to everybody's different well, but you'll see what's the same between all the different viruses, and that's their makeup. They all have some sort of genetic material, whether it be DNA or RNA, and that's they're going to make their proteins from that. Then they have the structure surrounding them. So they have this protein capsid that surrounds their genetic material. So all viruses, except for very few, there are some naked viruses that are just basically a piece of RNA. So we do know of a disease in a potato that's just naked RNA that causes a disease, which is kind of amazing that RNA can just survive on its own. But the viruses that we studied all have a capsid, and that capsid is made up of individual proteins. And then depending how that capsid is arranged, sometimes it's a helical structure. So when you look at the arrangement of the capsid in Ebola, it's a different arrangement to, say, the arrangement of a capsid in a polyhedral virus. So that would have been something that you would have seen. Of course, your viruses all have some sort of shape on the outside. We call them spikes, or they're proteins that allow that virus to attach to its host cell. And then, of course, then you have your complex viruses, which are your bacteriophages, which are capsid plus tail fibers and that. So those are even more complicated. I think we're all having virus overload with what's in the news at the moment with our wonderful COVID-19 that is a pandemic, and it's changed the way we all live. So very surreal time to be studying microbiology, but hopefully also understanding it. One of my students from last semester who had MERS as her virus emailed me to say, or actually she popped her head in my door about 2 weeks before spring break when it had just, that virus was just starting to show up. And she said, Dr. Berger, it's almost like my virus. What do you think? What do you think? And she emailed me yesterday, because she works in a hospital, saying, she said, I just want to check things up with you because everybody's asking me questions about this virus. And she said, and I know so much because I took microbiology. So thank you so much for everything you taught us. And then she's asking me for my opinion, how long it's going to last. And unfortunately, I think it's going to be here for a while, especially because people aren't listening. The other thing to understand with the viruses, that they basically go through a cycle. So they're not like a bacteria that just divides into two. They have a cycle where they have to attach to the cell, get into the cell, make more virus parts, and then get out of the cell. So whether you're looking at a bacteria virus or you're looking at an animal virus, they all have that same sort of pathway that they go through. Understanding the difference between lysogenic cycles and a lytic cycle. So that one is usually, that sometimes confuses folks. So try to think of lytic meaning lysis. So if you're in a lytic cycle, you're actively getting into a cell, making more viruses, and then breaking out that cell and releasing those virus particles. If you are lysogenic, you're getting into a cell, but then you're becoming part of the host chromosome, and you're hanging out there for a while before something triggers you, and then you go back into your lytic cycle. So kind of think of lysogenic as the lazy phase, where you decide to just hang out inside the cell until you decide to cause a problem again. We have the equivalent of a lysogenic cycle in human viruses would probably what we call latency. So let me just see if I can find my slide with, we talk about latency. So a latent virus is one that undergoes an acute infection, and then the virus never leaves you and it stays in your body and then comes back at a later stage. So this is usually when you're older, because when you're older, you are immune suppressed. And when your immune is suppressed, you end up being not able to keep that virus down. So that's why there's now, you might see out there, the zoster vaccine for people over 60 to basically just give their immune system a jump start again to make sure it still remembers chickenpox, because chickenpox is still out there. The reason you don't see too many chickenpox cases anymore is because people are vaccinated or they're immune to it. But what you do see, the varicella zoster virus coming back as a latent virus and coming back later in life. So other examples of latent viruses, does any of you want to jump in and tell me another example of a latent virus? So varicella zoster is a latent virus.
HIV.
HIV can be considered a latent virus. So HIV goes and hangs out and lives in your T cells, destroys your T cells, and then you eventually become immune compromised because you can't fight infections.
But shingles.
Shingles comes from, this is from the varicella zoster. So
I had it twice.
Sorry?
I've had it twice. It's terrible.
Yeah.
Yeah.
Not a good thing to happen. In fact, my middle son had chickenpox when he was about 2, and he had shingles when he was 8.
Yeah, I had, the first time I had them, I was 5, which was very unheard of. They don't, usually kids don't get them. And then I had it about 5 years ago, but it wasn't as bad.
So one of the things you probably want to do is get a zoster vaccine.
Yeah, I know. My aunt's been telling me to do it, and I keep forgetting to go and do it.
I think healthcare will pay for it once you're 60, so maybe you have to wait a while. I'm not going to get mine. And the other good thing is, if you were ever vaccinated against chickenpox, which now most of you young folks were vaccinated, never had chickenpox, you will never get shingles because you don't have latent virus living in your nerve endings. So you're okay. And then the other latent virus that I wanted to mention is herpes type 1 and type 2. So human herpes virus type 1, human herpes virus type 2 are also latent viruses. So you've all heard that term, herpes is forever. Well, that's herpes type 1 is the one that gives you the cold sores. Herpes type 2 is the one that gives you genital herpes, and both of those infections can keep reoccurring. And often when people have reoccurrence of those infections, something else is going on in their life, usually stress. Stress, sometimes sunburn for the ones on your mouth, can lead to those viruses. And as again, your immune system is not able to suppress it, and then you get an outbreak, then you go through your, basically that outbreak will cause you to get, ramp up your immunity to that again, and then you suppress it. So make sure you understand the difference between a latent disease and a latent virus. So the virus causes a disease. So just like when we talk about the TV is talking about coronavirus, it's actually incorrect. We should be calling it COVID-19. That's the name of the virus. Or they're calling it SARS type 2 because it's most similar to SARS. It's in the Coronaviridae family, and then it causes a respiratory infection. So that's why it's a bad guy, because those are the easiest ones to cause harm. Okay, so those are some of the biggies that I wanted to just point out to you from chapter 13. Anyone have any questions that you'd like to ask? Let me check my chat box for those poor people who didn't have microphones. Find the chat box. Okay, any questions? I hope Faith was able to come back. I can't see, I don't have my list anymore in front of me. Okay, so let's just quickly do a quick overview and cut some of the things for chapter 15. What are some of the things you should focus on for chapter 15? And then there is an online pathogen assignment for chapter 15. So chapter 15 is called Microbial Mechanisms of Pathogenicity. And this chapter ties in really well with chapter 16 and 17, because the reason pathogens make us sick is they are able to evade our normal immune system. They're able to breach us. So it's not like the minute this bacteria gets in you, you're going to get sick. It's got to have a way to get past your natural immune system. So probably think a really good focus is to focus on this slide here, because this is really your summary slide. And when you're answering your questions for chapter 15, which is coming up pretty soon, hope the date hasn't passed yet, probably by the end of the week. So you asked about your pathogen. What is its portal of entry? So portal of entry is, where does your pathogen get into the human body? And so when we look at portal of entry, the three main types are your mucous membranes, your skin, or the parenteral route. So when we look at mucous membranes, the most common mucous membrane for bacteria to get through, or pathogens, is your respiratory tract. And what are we seeing right now with COVID-19? How does it get into our bodies? It gets in via our respiratory tract. And that's why it's an airborne one. And the airborne ones are always tough. So whoever's bacteria is whooping cough, Borrelia burgdorferi, if I got that right, I know, whooping cough, I'm blanking on it. But anyway, whooping cough, respiratory tract affects those, the lining of your respiratory tract that's naturally preventing harm there. Gastrointestinal tract. So does your pathogen, you get it through food? Is it a foodborne illness? And is it able to survive the acidity of your stomach and get into your body via your gastrointestinal tract? So mouth, throat, stomach, small intestine, large intestine, and everyone out there are bacteria out there that each of them has their specific part. Or is it the genitourinary tract? Is it a pathogen that infects you through your urinary tract or your genitals, vaginally, or the male genital tract? So, and even if you think of some of the viruses that we've been studying, you can probably look at your assignment and realize that we have viruses that manage to breach those mucous membranes in all of those different places. So the one that comes for genitourinary tract, HPV, which I think wasn't that your virus, Ratana, HPV?
And yeah, that was mine.
Yep. Yeah. So that's its portal of entry. That's where it can cause infection. Think of conjunctiva. So if you've ever had pink eye, was it viral pink eye? Was it bacterial pink eye? And then your infection was in your eye. It was breaching your mucous membranes. Now, when we learn chapter 16, we'll learn about our mucous membranes are called mucous membranes because they are secreting mucus as a protection mechanism for us. And these pathogens, whether they're viral or bacterial or fungal, are able to breach those mucous membranes. So the most common mucous membrane is your respiratory tract. Then via your skin. So think of skin infections that come through your skin. Those are going to be those pathogens that are going to get through the pores in your skin. So anything that can actually breach your skin. So our skin is such an important part of our body. So keeping your skin healthy is so important because we know it's exposed to the outside all the time. And there are pathogens out there that are opportunists that can get in through your skin. So if you think of things like Staphylococcus aureus gets through your skin, normally it only gets through your skin when you've damaged your skin in some way. So maybe you have a rash, or maybe you've got very dry skin and your skin is chapped, so your skin is no longer protecting you. And then the third way, the parenteral route, is when you're basically going right through the skin and it's like getting to your bloodstream or getting underneath your skin very easily. And so that would be like if you have a deep cut or if you're getting a shot. If we're going to give you a shot or a vaccine, or we're going to use a needle, and that needle is going to have to go through the layers of your skin and make sure what we're trying to put in your body is past the skin. So parenteral is everything, ways you're getting past your skin, injected deep into your body. So that's why when you're having a surgery is always a problem for a parenteral infection, because you're breaching the natural barriers that you already have. And that's why for surgeries, we have all those sterilizing techniques that are out there. Okay, so after portal of entries, there's a couple of definitions that go with number of invading microbes and adherence. So obviously, one bad pathogen lands on your mucous membrane, it's probably not going to do anything. There has to be a certain number of them before you're going to actually end up with an infection. And then the other term, adherence, is how do you stick to those cells so that you don't just get naturally washed off. So if we jump into some of the parts inside the lecture, when we talk about adherence, are special structures that are on the microbe that are attaching to parts of our body or our cells to get in. So adhesion ligands are basically structures on that pathogen that are binding to receptors on the host. So what do we know about COVID-19? It binds to receptors that are expressed on the respiratory tract cells. And so those receptors are the ways that that pathogen sticks. And then once it's bound to that receptor, it can get in. So think of your pathogen. How does it stick to where it causes the infection? So if you had Streptococcus mutans, tooth decay, why is it able to cause tooth decay? Well, it produces glycocalyx, that sticky stuff. So it sticks on your teeth, and then it sort of makes that biofilm, and then your natural defense mechanism in your mouth, your saliva, and that is not washing it off. So what do? We brush our teeth every day to try and keep those bacteria from staying on our teeth. But even if you brush your teeth all the time, you're still going to end up with bacteria in your mouth. You never can completely get rid of it. The one I always like to talk about is Fimbriae, which is that structure on the outside of E. coli that allows E. coli to stick to your cells. So urinary tract infections are usually caused by E. coli getting in the wrong place, and the normal flow of your urine doesn't flush it out because of the Fimbriae that allows it to stick to those cells on the inside tract of your ureter or your urethra, and ultimately allows that bacteria to maybe even cause kidney infections. So that's why it can be a pathogen when it gets in the wrong place. And then, of course, why is all your skin bacteria not every time? Well, I had a shower today, I should have no bacteria on my skin. Well, no, because the skin bacteria that normally lives in that symbiotic relationship with us adheres to parts of our skin. So no matter how much you scrub your skin, you're still going to have some bacteria on your skin. And we don't want to scrub it all off because we know it is protective. So think about your pathogen in that, how does it adhere to your host cell before it causes an infection? Okay, so once that pathogen has adhered, how does it then penetrate the cell? So it's stuck on the outside. It's not automatically going to cause a problem. It's got to get inside the cell before it causes a problem. So that's where this question is really important. And that's usually the part of the question that students don't answer very well. So make sure you read up about it before you answer that question. And there's all these different ways that bacteria get into, or pathogens get into the cell, because outside of the cell or outside of the bloodstream, they're not going to be doing anything. So they have to have extra special ways to get in. So capsules. Remember what capsules are, not the capsule you swallow for infection, but capsules, when a pathogen can make a capsule, capsules are highly organized glycocalyx, and they prevent phagocytosis. So our normal immune system does phagocytosis, finds the shape on the outside of the bacteria that maybe it uses to adhere, but when it's already in our body, inside our bloodstream or inside our tissues, we've got fighting cells, and they are looking for those foreign shapes. But if you have a capsule, you're hiding those foreign shapes away, and it evades phagocytosis. So Streptococcus pneumonia, Haemophilus influenza, Bacillus anthrax all have the ability to make capsules, and those are pathogens because they're evading our immune system. So we will learn about phagocytosis in detail in chapter 16, but there's your first introduction to it in chapter 15. Other structures on the outside are cell wall components that some pathogens naturally make that also help them. Streptococcus pyogenes has something known as an M protein, which resists phagocytosis. Neisseria gonorrhea has a protein that's called an OPA protein, and that also helps it attach to cells and then grow inside the cells. And of course, if your pathogen is the mycobacterium, either tuberculosis or lepri, you have mycolic acid surrounding your cell wall, that you have that special cell wall component. And then again, you also are evading the immune system. You're not allowing phagocytes to digest you, or you're getting into the phagocyte, but because of that protective layer, you're now multiplying inside of the phagocyte. So your pathogens all have something unique to them that allows them to get into those cells. So whether they're cell wall components, maybe they're enzymes. So here's a whole list of enzymes. I don't expect you to know all of these enzymes, but think about your pathogen and does it have a special enzyme? So what have we learned over and over in this class? That bacteria secrete the enzymes outside to do their jobs. They're extracellular enzyme. They're secreted outside of the cell. So I have a gene to make that protein, and then that protein is an enzyme, and I send it out. And maybe it's a coagulase. It coagulates fibrinogen. It forms a clot, walls me off from the rest of the body so the host cell's defenses can't find me. Or maybe my enzyme is a kinase, which digests clots. So think about when you have when our body's protective mechanism is when you're bleeding is to make a clot. Well, there are some bacteria that can break down those clots, and that allows them to spread. So our body maybe forms a clot to protect it in a certain place, and then here comes a bacteria like Streptococcus pyogenes, produces streptokinase, which breaks down the clot. Or maybe you can produce an enzyme called hyaluronidase, which is an incredible enzyme because it breaks down the connective tissue cells. It breaks down the stuff that holds your cells together. So think of our cells are so well joined together to form that barrier. And now you've got a bacteria that can dissolve the stuff, the glue that's holding them together. And now that bacteria can get in. There's a really nice section in Mastering showing short little animations about all these different enzymes, which are really cool to watch because it helps you understand it. Or collagenase hydrolyzes collagen. And then, of course, this last one is a real stinker because IgA are our antibodies. So here are bacteria that have got an enzyme that destroys our antibodies. They destroy our first line. When we start making antibodies against them, they go, haha, I've got a weapon, and then they inactivate our antibodies. So this is why they're pathogens. This is why we get sick, because they have ways of getting into the right place and maybe inactivated some of our natural defenses. Antigenic variation is something that we see, especially in viruses. This is an example of trypanosoma, which is actually a eukaryotic pathogen that causes sleeping sickness. And this one keeps changing its outside proteins so that our immune system doesn't find it. It's like, okay, I've made an antibody to shape A. Now I'm making shape B. Okay, I've now made an antibody to shape B. Too bad, we're now on shape C. So it keeps evading your immune system because it's changing those surface proteins. And the best example of that are viruses. this is the influenza virus. Why we constantly need we need to update our vaccine to influenza is because of the antigenic variation, where it's changing the shape of the proteins on the outside. It's changing those spikes, the enzyme that allows it to get into the cell or the enzyme that allows it to get out. So you hear of H1N1 a lot. They're talking a lot about H1N1 because that was the influenza virus that was caused the 1918 pandemic flu. And antigenic variation is why we constantly to update our flu vaccine. Although latest work now, there's some really cool video out there. They're almost at the point where they might have found a shape in the influenza virus that we'll need to just get inoculated once, and then we'll be protected regardless of whether it decides to change its shape. So I was watching some of that over spring break. Other examples of antigenic variation, if your pathogen's Neisseria gonorrhea, it activates alternative genes for its OPA protein, that protein on the outside. So if you follow anything in the news, and this is something you can maybe talk about when you make your final disease presentation, that antibiotics that we normally use, which target these specific shapes on gonorrhea, are not working anymore because this bacteria is smart. Okay, so let me go back to that original picture. Okay, so when you're looking at evasion of host defenses, why did your pathogen get past our natural immune system? And then, of course, intracellular growth, where you actually, when the pathogen is maybe in a phagocyte, some pathogens grow inside the phagocyte. So HIV is an example of a virus that gets into a cell and then replicates inside, it hides away inside the cell. Mycobacteriums can do that. So you'll see that we'll talk about evasions of mechanisms of pathogenicity in chapter 16. So it's kind of, would be neat if it was, you'll see the tie-in once we've done chapter 16. Okay, next section in this chapter that's really important is how does it damage the host cell? So notice damaging of host cells is different from penetration or evasion. Once it's in the host, what does it then do? Why do we get sick? Because it's got to get to that part of the body before we get sick. And so this list here is what you'll be answering in your discussion of why it actually makes you sick. So let me find the list for damages to host cells. Okay, so there's your how you damage host cells. So number one way is taking away host nutrients. So it's in our cell and it starts stealing our food. It starts, using up our ions. So there's a structure called a siderophore. And siderophore are specific proteins that are secreted by bacteria, and these proteins steal iron, because all organisms need iron in some form for their metabolic pathways. So some bacteria have a structure called a siderophore that removes iron, and then some produce a toxin that causes the cell to release its iron. So I love this little cartoon here called Iron Thievery, stealing our iron, and it shows you different ways bacteria or fungi steal iron, and the different places in our body where we store iron, and then that iron actually gets taken. The bacteria wants to steal the iron by depending on which cells they infect. So if you have a bacteria that's infecting a certain cell, is the availability of iron there for it. So stealing host nutrients, iron is probably the number one of our inorganic molecules that they're after. Never mind our glucose and all our other stuff that we put in there. Okay, next one is causing damage at the invasion site. So cellular damage, so direct damage. So if you break open a cell like sometimes viruses do, you kill the cell. or you are producing waste products when that bacteria is growing inside the cell, or that fungus, their waste products is maybe toxic to us. Rupturing the host cells. So we see a lot of this, like if you think of fever blisters, when you see people, when someone has cold sores and those cold sores are broken open, all of those cells are actually destroyed. They're broken down, and that's why you can end up with those horrible, nasty blisters. One of the ones, if I have a picture here, but when measles, it might be in the virus lecture, but measles, when the measles virus infects cells, it bursts open the cells, but the nuclei stay in their nuclear membrane, and you end up with a whole lot of nuclei, and it's like a big blob of nuclei in one area. So you have this, and it's actually called a Koplik spot. So that's a very distinct diagnosis for a virus like measles. So how does it damage the cell? Disrupting nutrients, disrupting host function, rupturing the cell. And then, of course, number 4 is toxins. So toxins is just really a fancy word for something that is harmful to us. And so before we look at the direct toxins, if we go back to the beginning of the lecture, we actually talk about we talk about toxins way back in the beginning, about amounts of toxins. So toxins are proteins. So if you look at these structures here, those are proteins. And it's this protein that is toxic to us because it harms us in some way, because it damages something in our body. So Botox, produced by Clostridium botulinum, one of the most toxic substances known to man, 0.03 nanograms per kilogram can cause death. Sugar toxin, that's going to be a protein that's secreted by Shigella, causes terrible diarrhea. And we'll learn how that toxin, I think we've already talked about it, got given to the harmful E. coli. E. coli 0157H7 actually got the Shigella toxin by transformation, picking up the toxin from a dead Shigella cell, and now it's out there and dangerous. And then, of course, we talked about toxic shock syndrome due to Staphylococcus aureus. Toxic shock syndrome, that's the protein that is an exotoxin. So these are toxins that are made that are basically proteins that the bacteria sends out because that's what it does. It's part of its normal life. When it's growing and actively dividing, it's making these toxins. So when we look at toxins, we divide them into 2 groups, exotoxins and endotoxins. And we've talked about endotoxins a lot already in this class. Endotoxins are part of your gram-negative cell wall. Exotoxins are these metabolic products that are secreted by an actively dividing cell. Now, most exotoxins are gram-positive, most common are gram-positive, but gram-negatives can also secrete them as well. But gram-negatives are the ones that are responsible for your endotoxin because of lipid A, and hopefully we have heard this over and over in this course, that lipid A is the part of the gram-negative cell wall that when it's released, it creates a toxic effect. So some really nice slides in there just explaining exotoxins, that it's a normal protein made by the cell. Many are enzymes. What's the problem if you're an enzyme? You're soluble and you can be used over and over again, and that enzyme has some specific function for the bacteria, and unfortunately it has an effect on us. So toxins are often named for which cells they attack. So neurotoxins attack nerve cells, hepatotoxins attack liver cells. So Botox is a neurotoxin. Tetanus, the reason we have vaccinations to tetanus, we actually get a vaccine against the tetanus toxin. And that's why you need to constantly get that updated every 10 years. So when we look at exotoxins, there's 3 main types of exotoxins, and the AB toxin is the most common, how the toxin is made. So there you can see the DNA making the toxin. It's secreted in 2 parts, active and binding. The binding is going to bind to a receptor for that toxin, and then the A part is going to dissociate from it, and then it's going to have an effect inside of the cell. So most common way exotoxins work is this way. Other type of toxins are membrane disrupting toxins. So that even ties back in with how you get into a cell, damage in the cell. Maybe you secrete a membrane disrupting toxin. So what type of cells are you targeting? Are you targeting leukocytes? So then your toxin is a leukocidin. Or are you targeting red blood cells? Then you're a hemolysin. Or Streptococcus produces toxins called streptolysins. These toxins disrupt your phospholipid bilayer and break down cell membranes. So that's not only is it a toxin, but it's also the damaging effect on the cell. And when you're reading up about your pathogen, what way did your pathogen disrupt the cell? What kind of toxin did it produce? And then, of course, the third group of exotoxins are what we call the superantigens. And these ones cause an intense cytokine response in the host cell. Cytokines is just a name for chemical signals. And when you have superantigens, you can go into basically toxic shock. Fever, nausea, vomiting, diarrhea, shock, and even death. And this is the one that is toxic shock syndrome from Staph aureus, when young ladies use tampons incorrectly. And as I was sharing with some of the other day, I'm reading this book called Deadliest Enemies, very appropriate. And the author of that, Michael Osterholm, his early work was making the connection between tampon use and toxic shock syndrome, and how they were the ones doing these big epidemiologic studies of noticing the link between young, when people were admitted to the emergency room with toxic shock syndrome, how many of them were women, and they happened to have just had their periods in the last week of before they ended up with toxic shock syndrome. And they made the connection to tampons, and that's why tampons all have that great warning on them now. Okay, so this summary here is a great summary about exotoxins. The source, mostly gram-positive, relationship to the microbe, byproducts of a growing cell, chemistry, they're proteins made by, so you have to have a gene for that toxin, which then becomes the problem. Did you share the gene with somebody else? Don't usually cause fevers like endotoxins. They can be neutralized by an antitoxin. So an antitoxin is when you can make an antibody to that toxin. And that's why something like a tetanus shot is so important, because you get a vaccine to make an antibody to the toxin that causes tetanus. Your LD50, your lethal dose that would affect, that would cause 50% of the population die, is small. So that's a good thing. Well, no, that's a bad thing. You don't need a lot of it before it can be lethal. So that's why those Shigella toxins, and that you don't need a lot. And then, of course, this next slide here is showing different bacteria that make different toxins. So if yours is already in the list, you've already got information about the toxin that your bacteria makes. And here you can see what category they fall into. Is it an AB toxin? Is it a membrane disrupting toxin? What kind of toxin is it? Here's neurotoxins. Vibrio cholera is an enterotoxin. Staphylococcus aureus is that superantigen. And then the last part here, lysogeny. So remember, lysogeny is what I called, the way you can remember it is lazy, and that meant that this toxin is on a plasmid, and then it gets integrated into the host chromosome. And when it's on the host chromosome, it starts to be expressed, and the host is now making that toxin. So when a toxin is on a plasmid and can be shared between bacteria, that's a bad thing. And so out of this whole list of the ones you've seen, the only one that is not in lysogeny is the one that causes lockjaw or tetanus, Clostridium tetani. Here's that quick summary of endotoxins, relationship to the microbe. It's found in the outer membrane. If you don't know this by now, I don't know what I can do, because that's something you really all should know. It's a lipid A, and that's the problem, is if it's a lipid, we can't make an antitoxin to it. We don't make antibodies to lipids. We make antibodies to proteins. Its LD50 is relatively large, so you have to have a lot of it released before you're going to have that toxic effect. And endotoxins tend to cause fevers. So if you put this slide together with this slide, you have a nice summary side by side of the differences between endo and exotoxins. This slide is actually just showing how endotoxins actually lead to a fever, and how if that fever is not controlled, it eventually can maybe cause you to go into toxic shock. It can also do bad things like cause miscarriages. But remember, we have our own little immune system. We have cells that are going to be finding these bacteria that evaded our immune system. So if it happens to be a gram-negative bacteria, we're going to break it down by phagocytosis, and in breaking that down, we're going to release that endotoxin, which is going to get into our bloodstream, and ultimately it goes and has an effect on your hypothalamus of your brain and causes a fever. Fever is our body's natural response to fight infections, because if you like to be at 37 and I raise my temperature to 40, then you bacteria are not very happy. So fever is a natural defense mechanism, but we don't need fevers to go too high. So you can get endotoxic shock from gram-negative bacteria. This slide here is actually a way that we can find ways to look for endotoxins. It's a special assay. It's kind of a way to detect for the presence of endotoxins, because imagine this, you had a substance that you're going to use and you autoclaved it to make it sterile, but there happened to be gram-negative bacteria in it, and in the autoclaving you release the toxin. And so now that toxin is in what you think is a sterile substance. And so this is actually an assay, and sadly to use it, we kill horseshoe crabs. So very damaging to the environment. They say the horseshoe crabs survive, but actually horseshoe crabs, maybe 30% of them, when we bleed them for this specific substance, we end up killing them. We throw them back and most of them die. And there's a nice little scenario of an example of an eye surgeon where the patient had an eye infection, and the LA assay determined that there was an endotoxin causing the eye infection, because there wasn't a bacterial infection. It was actually the reaction was due to an endotoxin. So here's the last part of this. Here's showing those Koplik spots where you have what we call a cytoplasmic mass with all the different nuclei. And then the last part of this chapter is portals of exit. So most portals of exit tie in with the portal of entry. So if you have a respiratory tract infection, coughing, sneezing is going to spread it. COVID-19, respiratory tract spread by coughing and sneezing. Gastrointestinal tract, how are you going to be spreading that? All the way from your saliva to fecal matter. If it's a genitourinary tract portal of entry, same portal of exit. It's going to be excreted in the urine or vaginal secretions. Skin infections are not really going to be spread unless you go and physically touch that infection. So if someone has tetanus or if someone has diphtheria and you actually have a skin lesion, you're not going to see a problem unless you physically go and touch that person's infection and then put it in a place on your own body so it can get into the right place. So those are things that are not really that contagious. And then, of course, if you have an infection in the blood, sharing needles, syringes, or sharing it via arthropods that bite the blood. So maybe your pathogen was one, your virus was maybe West Nile disease that can be picked up by mosquitoes, or maybe your pathogen, your bacterial pathogen was Lyme disease, and then you get spread by ticks who are getting it via the blood. So again, when you're answering your questions for chapter, that last pathogen assignment, make sure you go and look at this thing and answer correctly. So these last two, pathogenic properties of fungi, pathogen, you can kind of go through them. We don't tend to focus on eukaryotes. We just don't have time. But the one eukaryote that we always want to talk about in Florida is red tide. And red tide is an algae that produces a toxin. It's called Carina brevis, and that toxin is a respiratory toxin. So that's why we end up with fish dying and other mammals dying on the beach. The problem with that toxin is it can bioamplify in the seagrasses, so that's why you end up with your manatees dying. And then, of course, there are other, if you like to eat shellfish, sometimes shellfish can be contaminated from algae that's out there. So those are things we have to watch too, especially living in a coastal resort. Okay. I'm exhausted from talking. Anyone have any questions about chapter 15 and your pathogen assignment? So ask some questions so I can stop talking for a while. But again, this is a great summary slide. And look at the part in your book that has the summary. Anyone, any questions?
I have a question about COVID-19.
Mm-hmm. Sure.
Have you heard about the malaria drug that they might be using for it, that they're going to test to?
Well, they're not going to use it yet, but one of the things they've noticed, because what they're doing is every single drug out there that's already been approved as a drug, there are labs that are testing it against this virus, because ultimately we want to be able to help those who get the infection. So the best way we're going to help this stop this virus spread is a vaccine, where we all get vaccinated against some part about it, and then we develop natural immunity to it. The vaccine isn't ready. There's trials going on already because clever scientists have already, racked up, but those vaccine trials will be very small until we know it's safe, and then they'll get bigger and bigger. The malaria drug, chloroquine, that you use against malaria parasite, it does seem to be that some labs have tested it, thrown it against this virus, and it does diminish it. Whether that is safe to use needs trials, needs to be tested. And they can't just suddenly, everybody just go start buying doses of that or getting prescribed it, because we don't have enough made. So we'd first need to prove that that is actually true before we would ramp up production. So if you follow Dr. Anthony Fauci, he was explaining that. So that's pretty much what's happening, is any drug that's out there. I've heard from my microbiology friends of two drugs, one that they used against Ebola, an RNA inhibitor that inhibits the RNA polymerase, seems to be effective against COVID-19. And the other one is a drug that we use for HIV infections, where you break down the capsid, you prevent the formation of the capsid in HIV infections, which we know now HIV is a viral infection that people can live with for a long time. Again, we can't just suddenly start prescribing that for everybody who's got a COVID-19 infection until we know for sure they're going to work. And we don't want to create a shortage for the people who are actively using that drug all the time.
I was mainly asking because my virus is coronavirus, the SARS, and I was reading an article about how it worked against, they used it for that, and how they're using it in China right now, and then they just gave a shipment to New York. They're going to start trying it this week.
Well, and I guess the people who are going to get that are going to be people who are already got
Yeah.
and they're in dire straits, and you know, we need you. You're on a ventilator already. Can we help slow that spread?
Yeah, because it's already apparently helped quite a few people that have gotten to that stage, and they've given it to them in China, and then some here, I think, too.
Yeah, I think we're really going to have to be thankful to the Chinese, because they were able to shut that infection down.
Yeah.
And they've also got good, clever scientists there, and this is a worldwide thing. We really need to help everybody. So yeah, all good things happening, and I'm glad you're asking those questions, because it means you're learning a lot of stuff just by doing a virus project, and then you can share that. But good information out there. Again, we live in a country where we don't just give drugs; they have to go through serious testing to make sure they're safe. And unfortunately, we're still in early stages. I just wish that we started worrying about this in January, when it first was showing to be pandemic, and started to be a huge outbreak in China. We lost 2 months in this country, and we're in a much worse place than we could have been had we started worrying about it in January.
Yeah.
So good question. So fun that you got SARS, which is the closest one to it. The other thing that you can do is antibody therapies. So we'll learn about passive acquired immunity in chapter 17, where when you've had an infection, you are making antibodies to that infection. So you have those antibodies circulating in your plasma. And what we can do is we can get your plasma from you, extract those antibodies from you that work for the virus, because you've made the exact one, and then we can do plasma infusions with people who are really sick. So that's what they did in the early days of Ebola infections, before, when the Ebola outbreak was so bad, and those definitely help. The problem with that type of therapy, it's incredibly expensive. If we can make a monoclonal antibody that will work against this this virus, that would also be really, really helpful. And we'll talk more about that in chapter 17, which is a couple of weeks away. That's something else to look out for. Okay, so good question. Anybody else with a question on chapter 15? So you all ready to go and answer that last pathogen assignment? And then once we've had our chapter, the next test, we will then go and you will move into the immunology section. So the other chapter that you had to just breeze through is chapter 20. And what I want to encourage you with chapter 20 is the only things you really need to focus on for chapter 20 are what is in the study guide. It's a 2-page study guide. And understand, when we talk about antibiotics, we're only talking about things that are naturally made by organisms against bacteria, and antimicrobials then includes all man-made drugs as well. So if you look at your study guide, there's a couple of definitions that you need to know. When we want to know the difference between broad and narrow spectrum, you'll need to understand that. Give some honor to Alexander Fleming, who discovered penicillin. These charts here are basically showing sources of antibiotics, and you see the names. They all come from microbes in some form. Maybe they were fungi, maybe they were bacteria, because bacteria, it's their own little defense system to prevent other organisms from growing around them. Hey, this is my spot. You can't grow here. I'm producing this, so you can't grow here. And then, of course, just go through the actions of them. So this picture here, how antimicrobial drugs work. And this clearly shows why these drugs do not work against viruses, because viruses don't have cell walls. Viruses don't have they don't make their own proteins. They don't have enzymes. So the only drugs, so when we look at drugs that we use against viruses, we use drugs that are going to maybe break down the cell membrane if they have one, or we use specific drugs that target virus replication. And that's it's not the mode of action of our antibiotics. And then moving through, just so this is a good chapter because again reminds you about differences between gram-positive, gram-negative cell walls, and then goes a little bit, just shows you how penicillins work, and then a couple of the ways you have antibiotic resistance forming. So this is great activity. We talked about this when we did bacterial genetics, is how many of our drugs act right at the inhibiting protein synthesis. So blocking the formation of a peptide bond. So actin over here, actin over there, preventing the formation of the peptide bond when you're making your protein, or preventing binding of your messenger RNA on your ribosome, or messing with the binding of transfer RNA. So all of those are actions that inhibit protein synthesis, blocking the channel that the protein can't come out. So drugs that inhibit protein synthesis are going to be basically broad spectrum, because all bacteria do protein synthesis, and they're not bactericidal. They don't kill the bacteria, they just slow down the growth, and then your immune system is going to take it up. So these would be very important to use against gram-negative bacteria. So don't get bent out with the names of the drugs, rather just have a basic understanding of how they work. And then, of course, the last part here is talking about resistance to antibiotics, how bacteria are becoming resistant. So again, there's a video that was when we're looking about how you kill bacteria. That was the guy who talked about transformation, transduction, and conjugation. Those are all the ways bacteria can share resistance to antibiotics. So this is also a picture that is in your study guide that you want to answer. And then also there's a little bit of information. We did this in class where we did the Kirby-Bauer test, where we figured out which antibiotics were effective against certain bacteria. So there's your zone of inhibition. And then how much antibiotic to give. So we always want to know, we want to give the lowest dose of a drug that's going to work. We don't need to give the highest amount if it's going to work. So there's specific tests that have been done on that end. So they do things like broth dilution tests to know what is the exact amount of that drug that is effective against that bacteria. And then synergistic or antagonistic is another term that you learn. And then, of course, resistance. I love this cartoon, so make sure you look at that cartoon. And sharing resistance. So here you see E. coli with a plasmid that's got antibiotic resistance. And now after conjugation with Salmonella enterica, which is a very similar bacteria to E. coli, both live in your gut. And now you can see that it's actually passed that plasmid over. So these are ways bacteria can become resistant by being around other bacteria. And unfortunately, we're running out of antibiotics. Hopefully this pandemic with COVID-19 is going to wake up the world to the other important problems we have with regards to microbes, and antibiotic resistance is one of a huge one. So any questions for chapter 20? I kind of rushed through it a bit.
I had a random one. So, and I don't know if this would apply to this chapter, but when, like, say you have a UTI and they send it out to lab and they give you an antibiotic, but then they call you a couple days later and say, oh, you need a different antibiotic because they're seeing what strain it's resistant to or whatever. Mm-hmm. What happens if you ended up with a gram-negative thing or whatever, and they gave you an antibiotic and it killed it, and now you've got the toxins in you, and then they're like, oh, you need something different? Sorry.
It's okay. This is cool. You see your family? Oh, there's your dog that ran away. Is that the dog that ran away?
It's my dog that ran away. He's here.
So that's a really good question, because what happens is when you go to the hospital and you've got a and you've got a UTI, they basically prescribe you a broad spectrum antibiotic in the hopes that it'll work. And then if they send your urine out for testing, then they can actually test what is most effective against your bacteria infection that you have, and then that's why they change the antibiotic. But unfortunately, you've already been taking that one antibiotic, and that always bothers me, because what effect did that other antibiotic have on my cells while you were trying? But ultimately, the doctor's goal is to get rid of the infection, and this is where we live in a society where doctors overprescribe antibiotics. And again, depending how sick you are or how quickly you want to feel well, if you don't want to wait, doctor doesn't want to send you away with nothing when you've obviously got a UTI, because they're worried about their butt, getting sued. Oh, I had a UTI, my doctor didn't do anything for me. So they're always going to probably prescribe a broad spectrum in the hope that it works, and then maybe they figure out, oh, she's got a really nasty other bug. Well, we'll add another antibiotic on top of that. But in the meantime, maybe some other bacteria became resistant to that other bacteria that the other antibiotic that you got. So it's scary, and those are things that we need to get better at. One of the things that I talked about way back in was it chapter 6 about quorum sensing, how bacteria talk to each other. So if you have time, because you all have so much time, I'll put the link to the TED Talk again. Bonnie Bassler talks about quorum sensing and blocking quorum sensing, blocking signals between bacteria so they don't launch the pathogenicity. So, when we talk about toxins are released by, you're only going to get sick when you have thousands of those toxins being released by a big enough sample of bacteria. If you just had one bacteria in your system and it said, I'm going to make this toxin, it's not enough to make you sick. But if you have a dose of maybe a billion bacteria and they're secreting that toxin, then there's a greater chance you are going to get sick. So can we block that quorum sensing so they never ever get to the point where they say, oh, time to release our toxin? Or there's enough of us growing in enough samples. So lots of good questions out there about that. So, but yeah, try keep minimum antibiotics in your kids. And then, of course, the whole big thing of the food industry, antibiotics that are in the food industry. We have huge amounts of antibiotics being given in the food industry to our pigs, in farming systems, which now there are big movements to block that. So it's been banned unless it's been prescribed by a vet. So what do farmers do who are used to always keeping their animals on antibiotics is they just have the vet do a prescription for their herd or whatever they're doing. But we need to move away from that as well. So antibiotics were the miracle drug of the 20th century. They're not going to be the miracle drug of the 21st century, just like pandemic viruses are our new normal, which we're all dealing with right now. So, good question. Anyone else have a question? Well, I didn't think we would be here for so long tonight, but good staying power, folks. So if you don't have any other questions, remember that there's a link in announcements for online office hours. And those are times I'll definitely be online, but if there's any other time that you want to meet with me and ask me questions, then feel free to jump in. Next week, your next test will be an online test for next week. So you don't have anything new to learn this week. So go back and review everything from 10 through 15. And then the online test we will open next Monday after we finish having a review session. So next class Monday night will be any review questions you may have from 10 through 15 and 20. And then after we've closed that review session, then I'll open the online test for 24 hours for you to take. And if that time doesn't work for you, then I will can go and give you another time to do it in. So that is a possibility that I can do. Okay, So this Zoom meeting is so long. When I upload it, I'm going to upload it in 2 segments. One segment is going to be for the labs, and the other segment is going to be when we've kind of talked about chapter 15 and 20. So you can home into different sections just to make it easier for you if you want to go back and review. But I mean, you have all the lectures online and they're broken down into segments too, if you want to go look at that. Okay, so thanks for joining me. We had 19 people who started. We're down to 15. So I don't know what happened to the other 4, but hopefully they're okay. And I didn't see anybody trying to come back in. So, and I'm going to email those students who weren't able to come tonight. So stay in touch. Keep in touch with your friends you made in class. That you all need each other. You're not isolated. You're connected by Canvas. So stay in touch with your friends that were at your table. I'm sure they'll be very happy to hear from you. Okay, so good night, everybody. Thanks for joining me tonight. Bye-bye.
I have a question about the exam.
Yes.
When we take the exams, are we still gonna have like the summaries like that we have to do like at the end, or is it just gonna be like multiple
You mean no? Yeah, you will have some short response questions where you type in some answers. So they will be just quick short questions. So you want to be prepared for that, and that's the way you can earn points. So pretty much the same thing. And I also have some bonus questions in there, like I always do. But again, we're gonna just want people to be honest as best they can.
Yeah.
Not using notes, go through it. So each question, the way I'll set it up, that the question, you'll only get to see your question 1 and then it'll lock. So you'll answer question 1 and then you move to question 2 and it will be timed. So you can't go look up the answer and then come back because you will run out of time. So that's the only way I can avoid people doing that, is making, locking the question and then shutting it down. So
how long is it going to be? Like 60 minutes?
Probably about an hour. But most of you, if you know your stuff, you'll get it done in 30 minutes. When I've done online tests, the students who know what they're doing, they're done in 30 minutes. And the ones who use all the time are the ones who don't know their stuff. So study hard in the next week. Get your pathogen 5 assignment done. Make sure you reply to everybody else so you see the differences that we have between how your pathogens are different, how other people, what toxins they make, their portals of entry, their portal of exit. So all of those will help you see that bacteria all are different and they're pathogens because of those things that they can do. And stay safe and social isolate. Stay away from other people. The sooner we all social isolate, the quicker this infection is going to end. We can go back to normal. And for so many people, it's a real hardship because you don't have jobs anymore. So let's hope the government comes up with a good plan and we live in a country where they can give money to us people. I'm okay. They don't have to send me a check, but I know many of you need a check from the government. I'll need a check if they close fall semester down. So, okay. Nice to see you all. Stay safe. Stay well. And if you do get sick, let me know if I can help in any way. If something comes up and you're sick and you can't, you're not going to be able to take the exam next week. Hopefully that doesn't happen, but let me know. I'm here to accommodate whatever you need. If you need extensions, let me know. I'll give you extensions. You just got to be done by April 29th. Okay? I have to have grades in by April 29th. But if you need any extensions for some reason, just let me know. Okay? Take care.
Thank you. Thank you.
Good night.
Good night.
Good night. Bye.
Bye-bye.